Recording is about to start. And hopefully I'll see the blue bar. Where's the blue bar? Yes, there it is. Wonderful. It is recording. And Bernardo, as usual, thank you very much. That's great. Screen sharing. And let's hope I can do the screen. Where am I? I'm here. Right. Now, big question. Can you see my screen? Everything all right? Yes, brilliant. Wonderful. So, technology works. It's all good. Yeah. And here, obviously, you see the topic that we are going to do today. Again, we've got quite a lot of ground to cover. Is it blue? Yes, it is blue. Mind you, I'm pretty colorblind, I have to say. Very much to the amusement of my wife when I pick socks. And sometimes she says, no, you can't do that. You can't wear a pair of blue, one blue and one black sock. I mean, to me, they are all black, but obviously to her, they are different colors. But then I was told that guys only do the computer colors, the RGB, red, green, and blue. And, you know, we don't really bother with all the sort of nuances, aubergine, and mauve. It's red. Either it's red or blue, right? Make up your mind. That's at least that's how I work. So I'm glad I'm not the only one. Right, now shall we do a gentle start to the topic today? And the topic is absolutely right. Socks is socks. This is the stuff that you put on your feet. Don't even worry too much about the color. The purpose of a sock is to keep your feet warm and stop you from getting blisters. Full stop. End of. Doesn't have to look, you know, great, sexy, or God knows what. It's just sock is sock. Absolutely right. Okay, so today we are going to discuss a topic called gluconeogenesis, and also how we can use amino acids as a fuel. Now, just a quick revisiting of what we have done so far. We looked at the fuel availability for different organs, and you've seen this table before in one of the sessions where we looked at, for example, the resting state. We looked at short-term starvation, just for a few hours, or when the muscles are very active. And then we are looking at long-term starvation. So several days, weeks, maybe even months in terms of starvation. And we looked at the requirements of the organs. So we said that the brain predominantly uses glucose. Also, you know, short-term starvation, a few hours, we need glucose. but the brain then can switch to a different fuel form, which we've discussed on Wednesday, and we can supply the brain with the ketone bodies, which can replace the glucose. But in the interim, we really need to provide the brain with some glucose, because the ketone bodies the brain adjusts only after 2 to 3 days. We can't keep the brain hanging for that long. Skeletal muscle, not a big problem, because we've got large glycogen store. We can use glucose. The muscle can use fatty acids, can use ketone bodies. They can do basically whatever they want. And long-term starvation is not a problem. Likewise, the heart muscle, the adipose tissue uses also fatty acids and so on. Kidney uses, if available, glucose, but it can use ketone bodies from fatty acids, so not a problem. The liver usually doesn't use glucose anyway, so it's not a problem. It uses alpha keto acids, and it maintains that because the liver doesn't use the fuels that it will provide for other organs. And that's quite an important thing to remember. So far, so good. But we have one problem, one major problem. And these are the erythrocytes. These are red blood cells. Red blood cells definitely have no other option, but they need to use glucose. They do not have mitochondria because, and therefore they can't use fatty acids or ketone bodies. Their only food source is glucose. So this is a problem, isn't it? Because if we are going into starvation mode, we do not have glucose left. So there is no glucose. So how on earth are we going to feed our oxygen transport system, our red blood cells? Now, what we can do is we can sort of switch on an emergency supply. So emergency supply of glucose. And this is exclusively for red blood cells. All the other organs that we have can switch to other fuels. Red blood cells simply cannot. They must have glucose, otherwise it's just game over. So where do we get glucose from? How can we provide the erythrocytes with glucose? And there's one very important thing that we need to remember. Animals, like humans, animals can not produce glucose from fatty acids. It's not possible to convert fatty acids into glucose. Simply doesn't work. Now, you've probably noticed that I've written animals because plants can. Plants can actually use fatty acids and produce and use these fatty acids, break them down to acetyl-CoA, and from the acetyl-CoA sort of, they can produce glucose. Most fungi can do that. Many species of bacteria can do that, but animals can't do it. We cannot use fatty acids to make glucose. So we need glucose for the red blood cells. So how can we do it? Well, We know that many steps in glycolysis, so here this is again glycolysis, and many steps in glycolysis are, of course, plants are superior. There's no doubt about it. Plants don't need vitamins. They produce everything themselves. They're just truly amazing. Anyway, in glycolysis, many steps are operating at a delta G of around 0-ish. This means that many steps are reversible. So in theory, we could run basically glycolysis in the opposite direction. So if we have, for example, pyruvate, the idea would be to run glycolysis in the opposite direction and end up with glucose. At least that would be sort of a nice thing if we could do that, because then we could produce glucose for the erythrocytes. There is actually one problem. Actually, it's not one problem, it's 3 problems. 3 problems that stop us from running glycolysis backwards. And these 3 problems that we have are 3 irreversible steps. 3 irreversible steps in glycolysis. And that stop us from running it backwards. So let's see if we can find these 3 irreversible steps. Let's take that away. Let's take this away. So where do we have them? The first irreversible step is here from glucose to glucose 6-phosphate. This is an irreversible step, so we can't use the same enzymes for that. The second irreversible step is here from fructose 6-phosphate to fructose 1,6-bisphosphate. And again, this is an irreversible step that we need to sort of be creative and try to overcome somehow if we want to run it backwards. And the final step is from pyruvate here to phosphoenol pyruvate. That's the final step. So 1, that's the first step, that's the second irreversible, and that is the third irreversible step. Now, if we could overcome these irreversible steps, then it would be not a big problem to run glycolysis in reverse and actually produce glucose. So let's do it in reverse order. So let's look at the last step, at the conversion of, so that would be the third irreversible step, at the conversion of pyruvate, so COO minus COCH3, and we want to convert that into phosphoenol pyruvate, which is usually abbreviated as PEP. And this guy looks like COO minus what is it? We've got an H here. O phosphate. No, we don't have an H here. Sorry, I'm lying. And we have a double bond CH2. We can't actually do this direct step. This is not possible. but we can do a little bit of a bypass, if you like. What we can do is we can use ATP. It's converted to AMP, if I remember correctly. And we can use carbon dioxide. And we can use plus biotin. This is a cofactor or coenzyme, what people sometimes call. This is also known as vitamin H or vitamin B7. It's also known as. And we can add this carbon dioxide to the end of pyruvate. And that would happen inside the mitochondria. COO minus, CO, CH2, COO minus. So we added basically the carbon, and you're absolutely right. This is oxaloacetate. Oxaloacetate. Absolutely right. So we can do that. So we've got oxaloacetate.
So long.
And what can we do now with it? Now we can transport the oxaloacetate out of the mitochondria. So here that was all in mitochondria. In mitochondria. We then transport it out. How do we actually transport oxaloacetate out? Yes, it is in the matrix, absolutely right. We have to use this malate shuttle that I've introduced earlier. So we have to convert it to malate, transport it out of the mitochondria, and convert it back to oxaloacetate. So back to oxaloacetate. A bit of a detour, but it's not that big of a problem. So we've got oxaloacetate there. And then we can again use ATP convert that to ADP, and actually form from this oxaloacetate, we basically chop off the carbon dioxide that we just added, CO2. We lose that again, the carbon dioxide, and we get our phosphoenol pyruvate. This is the enzyme PEP, phosphoenol pyruvate carboxykinase. And this enzyme here is the enzyme pyruvate carboxylase. It's all very confusing. I do not expect you to learn these enzymes. The point of converting it from oxaloacetate into malate is because mitochondria have no way of exporting oxaloacetate itself. They have malate transporters, but they cannot transport oxaloacetate in any shape. So therefore, you need to convert the oxaloacetate into malate that you can transport. And when you are then in the cytoplasm, you convert the malate back into the oxaloacetate. So that's sort of just a transport form. Yep, does that make sense? And now, when people said we need to learn from plants, here's something really, really interesting. Because you always learn that plants can fix carbon dioxide, can't they? Plants fix carbon dioxide. And that's certainly something that's absolutely amazing. No, it prevents the oxaloacetate from leaving the mitochondria, usually. That's the reason why there are no oxaloacetate transporters. But the malate can go out. Now, plants can fix carbon dioxide. Can we fix carbon dioxide? That's exactly the one. You're absolutely right. You heard about the malate shuttle with the NADH. Absolutely right. That's the same thing. But here in this case, we don't focus on the NADH as a transport. Here in this case, we focus on the transport of basically the oxaloacetate out. Absolutely right. Now, we know that plants can fix carbon dioxide. Can we fix carbon dioxide? Absolutely right. Yes. With the help of the biotin here, with this vitamin H, we do fix carbon dioxide because we add a carbon dioxide actually to this pyruvate. So we have added carbon dioxide and we fixed carbon dioxide. With the help of this vitamin H, B7, we can fix carbon dioxide. So we can basically do the same things as plants. Isn't that amazing? We lose then the carbon dioxide again, but that wasn't the question. We still can fix carbon dioxide. We have this ability. So the first irreversible step, we needed to go from pyruvate to phosphoenol pyruvate. So basically, tick box. Hey, we've done that. The first hurdle is taken. Now, where's the next one? Yeah, the next one is here, 2. So we need to go from, that's the second step, fructose 1,6-bisphosphate. We need to go to fructose 6-phosphate. We actually lose a phosphate residue. The reverse reaction is catalyzed. Where is it? The reverse reaction is catalyzed by phosphofructokinase 1. So how does that work? This is an irreversible step. Again, like the one above. So how is that catalyzed? Actually, what happens is, if I draw that out, fructose 6-phosphate to 1,6-bisphosphate. We said this reaction is catalyzed by the enzyme PFK1, phosphofructokinase. But we need the reverse reaction, and this is catalyzed by the enzyme fructose 1,6-bisphosphate. bisphosphate phosphatase. This is how this one is catalyzed. So this reaction here by PFK1, that's an irreversible step. But this one here is then catalyzed. We just simply cleave off the inorganic phosphate. Absolutely right. Now, you see, can you remember actually what this is called? We have, on one hand, we have phosphorylation, and on the other hand, we have dephosphorylation. And it happens in the same location. In the cytosol. Thank you very much, Adam. Absolutely right. We have zero order ultrasensitivity because all we need to do is just simply change the, yes, absolutely right, is we have got a switch here. A very, very sensitive, very, very sensitive switch. Very sensitive switch that we have. So if we increase the activity of PFK just a little bit, we go into this direction. We go into glycolysis. If, on the other hand, we increase the activity of the phosphatase just a little bit, then we go immediately into the opposite direction. To the opposite direction, into gluconeogenesis, into the new production of glucose. So just by changing these properties of PFK1 and the phosphatase just slightly, we switch the pathway from either going into glycolysis into glycolysis. So either into glycolysis or into gluconeogenesis, just by slightly changing that. And we've discussed that, for example, PFK1 is activated by AMP or fructose 2,6-bisphosphate. And likewise, we have activators for the phosphatase. So phosphatase would be, for example, activated by levels of ATP. So if we've got enough ATP, this works quite nicely. Why is that important? I forgot to mention this first pathway here, this what I called 3, it's very, very costly. If you look at that, in this conversion from pyruvate to PEP, we need ATP for that. So that's quite costly. We can run gluconeogenesis only if we've got ATP. And the sensor that we have ATP is here, these phosphatase. We also know that PFK1 is inhibited by ATP. So this was the second step. We have this switch-like behavior in the same location. So, hey, we are on the way. We've done 3, we have done 2. We just need, we just need to do 1. So 1 is the conversion of glucose to glucose 6-phosphate. So that's 1. So we have glucose to glucose 6-phosphate. And now this is an irreversible step. And we need to figure out how we can actually do this step here. So this one was catalyzed by hexokinase. And this step here, glucose 6-phosphate to glucose, is in principle the same as we have seen for the phosphofructokinase. We do a hydrolysis reaction and we use the enzyme glucose 6-phosphate phosphatase. So that's in principle the same reaction as we have in step 2. Looks similar, like zero order ultrasensitivity, switch-like behavior, only with the really nice effect that in this case we have different compartments. Different compartments. This process happens in different compartments. Where would we find hexokinase? Where would we find the enzymes for glycolysis? In the cell. In the cytosol. Absolutely right. We find glycolysis in the cytosol. That's right. So this part here is in the cytosol, whereas this part here actually, and Cami is right with that, happens in the endoplasmic reticulum. The enzyme glucose 6-phosphate phosphatase is an ER protein and can only work in the ER. So we have spatially separated the phosphorylation reaction from the dephosphorylation reaction. And I think that's, again, is quite interesting. And it gets even worse because only a few organs express this enzyme. Only a few organs produce this glucose 6-phosphate phosphatase. This is the liver. We find this enzyme only in the liver and in the kidney. And I think there are some other very specialized cells. I think some astrocytes can do that as well, can express this enzyme. But not normal, for example, muscle cells would not express glucose 6-phosphate phosphatase, and therefore only liver and kidney actually can do gluconeogenesis. Gluconeogenesis. All the other organs are consumers of glucose, but liver and the kidney can actually produce glucose. And however, the problem is still, if we want to produce, so production of glucose requires ATP. We require energy We require energy to produce glucose. That is what the definition is of gluconeogenesis. Glucose, neo, we produce it from scratch, and genesis means production. So glucose new production, that's glucogenesis. We require ATP for that. Absolutely essential. And usually people say it starts with pyruvate. So that's the starting point of gluconeogenesis. I personally prefer saying that it really starts with oxaloacetate, because that's sort of that is where the whole thing starts. And the pyruvate that we use just is used to sort of generate the oxaloacetate. But in general, people say it starts with pyruvate, or the reduced product. It could also, the cells could also use lactate. So we require ATP. So ATP cannot be gained. And you're absolutely right, Reynolds. It is not cost effective. We need quite a significant amount of ATP to produce the glucose. So why on earth do we do that? Why on earth do we why on earth do we spend energy to produce glucose? What's the point? Why do we produce glucose? But if it costs us. Absolutely right. We need the glucose, cost it what it might, to keep the red blood cells alive, and also to gap this transition from the glucose to the ketone body switch in the brain. Absolutely right. It costs us energy to make this glucose, but it's absolutely essential, because otherwise the red blood cells would die. That's why we have to make an investment of ATP to produce glucose. Does that make sense? So what are we then doing if we, let's go back to the table, so we keep everybody happy and we can survive long term just by tapping into the fat stores. But what happens if we run out of fatty acids? No more fatty acids. We are running out. Last resort, Charlotte, you are absolutely right. We are going to use amino acids. That is what we have left. And with the amino acids, if you fast for, say, 3 months, there is no fat left. Trust me on that. So with the amino acids, we have basically, so let's say we have amino acid. We have 2 different things that we can do with amino acids. What have we got with an amino acid? We have a carbon skeleton. carbon skeleton. So if we just simply have an amino acid like COO minus, C, N, NH2, CH3, so that would be, what amino acid is that? Alanine, yep, well done. So that's the amino acid alanine. And what we need to do in this case, we need to get rid of the amino group. So what we do with this, how do we do that? Well, this reaction again happens predominantly in the liver. And you see the liver is really the hub, the metabolic hub, with the help of yet another vitamin, and this in this case is called pyridoxal phosphate. Pyridoxal pyrophos. Is it pyrophosphate? Phosphate. No, phosphate. Pyridoxal phosphate, or also vitamin B6. We take the amino group and convert this molecule to COO minus COCH3. We also use, or we generate NADH in this case. What is this molecule here? What is this molecule here? So here we have alanine. Yes, we are back to pyruvate. That's pyruvate. Absolutely right. And what can we do with pyruvate? What we just discussed. We can make glucose. So we would call this amino acid as glucogenic. That would be a glucogenic amino acid because we can make glucose from it. So there are other amino acids that would break down the carbon skeleton, that would break down, and I should spell that with a K, skeleton, can either be used to make glucose, so that would be glucogenic, or we can, very good question, and the answer is no, we cannot convert every single amino acid into pyruvate. Some of them can only be converted into H3CSCA. What is this? What is this molecule? Yep, absolutely right. That's acetyl-CoA. But what can we produce from acetyl-CoA? We said the pyruvate produces glucose. What do we produce from acetyl-CoA? We could put that into the Krebs cycle, yes, but we could also use it for the production of something. Ketone bodies, absolutely right. So we can produce this into ketone bodies. And therefore, these amino acids are called ketogenic amino acids. These are the ketogenic amino acids. There are only 2 amino acids that are purely ketogenic. These are K and L. These are the 2 exclusive ketogenic amino acids, so lysine and leucine. There are some amino acids that are purely glucogenic, and there are some amino acids that can do both. Right? So that would be the fate of the carbon skeleton. We can use, under extreme starvation, we can use the carbon skeleton after we've removed the amino group. We can use then the carbon skeleton either to produce glucose or ketone bodies, right? Now, what are we going to do with this amino group? So we have amino acid, and we remove the amino group. And here we would get actually this would be called an alpha keto acid. That's the alpha position here. Alpha keto acid. Actually, if you remember, this is the food that the liver uses. So we converted the amino acid into an alpha keto, oops, that looks interesting, into an alpha keto acid. But what are we going to do with the ammonia? Or in this case, it would be ammonium. What are we going to do with that? Well, what we can do is we can transfer that onto another alpha keto acid. And make another amino acid, a different amino acid. Pyruvate is an alpha keto acid, absolutely right. So this reaction is called transamination. Transamination reaction. That's the one. Yep. And that is basically the fate of amino acids in the liver. They become transaminated, so we basically can produce whatever amino acid we want if we have the original, by just simply transferring the amino group to somewhere else. So that's a transamination. And the transamination reactions are really sort of, they link the amino acid metabolism linked to the glucose metabolism. They are the big link, this transamination reaction, between these 2 very important compounds, amino acids and glucose. This is the link between them. But, you know, one of the problems that we have is at one point we need to get rid of the amino groups. And the way we can do that is we can transfer the amino group onto alpha ketoglutarate. We transfer that onto it. Well, let me draw this nicer. So that would be another transamination reaction, if you like. And we get another amino acid, we get glutamate. Right, so we produce lots of glutamate. What can we do with that? Well, the glutamate, we can take the glutamate, remove again the amino group, this time as NH3. And that is an oxidative process, which is catalyzed by the enzyme glutamate dehydrogenase, TH. And we also gain NADH. Glutamate dehydrogenase is a really weird enzyme. Yes, transamination occurs in the liver, pretty much so. Glutamate is a really weird enzyme because it can use NADH, or NAD, I should say, NAD plus. But it can also use NADP plus. And that's really something that doesn't happen very often with enzymes. So we can oxidatively remove the ammonia here. But, you know, what do we do with the ammonia? Here comes one more cycle that we use in the body, and that is basically to get rid of NH3. We combine NH3 to NH3 plus 1 carbon dioxide plus ATP. It costs us. The waste disposal is costly. This produces this compound NH2, NH2, yeah, CONH2. This is urea. And that is what we are going to secrete. There is a cycle. This is called the urea cycle, which uses a number of components. I'm not going to expect you to know the details of the urea cycle, but that basically takes the amino group, eventually it takes the amino group and converts the very toxic NH3, the ammonia, and it converts it into the less toxic urea. And that is what we secrete. At least that is in most mammals most mammals. Other organisms, like for example birds, have a different way of getting rid of the ammonia. And in these organisms, like birds and reptiles, instead of urea, they excrete they excrete uric acid. So that's their way of getting rid of things. For urea, you need quite a lot of water for the excretion, whereas for uric acid, you need to invest less water. So that's why when you want to save water, these organisms secrete the ammonia as uric acid. So basically, what we do is we take an amino acid, we remove the amino group, use the carbon skeleton either to produce glucose or ketone bodies under extreme starvation. That's the carbon skeleton. The amino group then is transferred onto other acids, alpha keto acids, in this transamination reaction. And ultimately, it is taken then onto glutamate. The glutamate then is used to again remove the the ammonia. So we go from glutamate again to alpha ketoglutarate. We remove the ammonia and go into the urea cycle, and eventually we pee out the waste nitrogen. We do it as urea. Birds and reptiles do it as uric acid. Does that make sense? Good Lord, it's 2 o'clock already. Any questions? If there are no further questions, I just say have a lovely weekend. We covered a lot of stuff this week. Next week will get a little bit easier. And next week we only have the Wednesday session. There's no session on Friday because it's Good Friday. So thank you and have a lovely weekend. Be good. Don't do anything I wouldn't do.